Hello students, now we will discuss about Porifera. Porifera is the phylum in animal kingdom and commonly called as sponges. These sponges, they are both marine and freshwater. They are both marine forms and freshwater forms. Under marine, we can take example like cycon, Leukocylenia, and also sponges. Under freshwater form of sponges, we can take spongilla. Cycon is also called as scypha. Sponges or spongia is also called as bath sponge. Only one example is under freshwater forms, that is about spongilla. Next, another example is also there, that is Leukocylenia. Basically, most of the porifera or the sponges are acoelomates. Mostly, sponges are acoelomates, means there is no coelom. But cycon and Leukocylenia, these both are exhibiting radial symmetry. But cycon and Leukocylenia are of, or they have radial symmetry. Sponges are sedentary or sessile, means they do not move from one place to other. They are sticked on to one place at one substratum, means they are not moving organisms. Sponges are sedentary or sessile. Sponges are considered as primitive multicellular animals with a body plan of cellular level of organization. Cellular level of organization means there is no coordination between the cells that are framing out the sponge's body. Each and every cell is doing its own specific function. One cell is not related to other cell in metabolic activities. Next, sponges have water canal system or water transport system. It means the entry of water into the body is by one way and exit of the wastewater or the used up water from the body is from other entry. So we know that meaning of a canal is from one side to other side. In the sponges also it is same. So in sponges exhibit water canal system, which is also called as water transport system. Here we will write just a meaning of this. It means water enters into body by oval shaped cells, ostia, and goes out of body by one by other entry or other opening called osculum. Osculum is also considered as anterior opening. These ostia are also termed as minute pores. Next. From osculum, water enters into spongiocele. From spongiocele, water enters through, means out of the body through osculum. Next one. This, what is use of this water canal system? It helps in gathering of food, respiratory exchanges, and removal of waste water. So, use of water canal system in sponges is gathering of food, respiratory exchanges, and removal of waste from the body. These ostia, they are made of tube-like cells called as porocytes. They are made of tube-like cells called porocytes. These ostia or the porocytes, they help in regulating the flow of water into the body, means into the spongiocele of sponges. They also regulate water currents. Sponges are solitary, means they live in single or in colony form. Solitary or colonial. In the anterior part of the body, sponges have a paragastric cavity which is called as spongiocele. Next. Spongiocele is lined by choanocytes. Is lined by choanocytes. These are also called as collar cells. These are also called as choanocytes, are also called as collar cells. Body of sponges have a skeletal form which is made up of spicules, supported by skeletal cells called spicules. Body wall consists of outer pinacoderm and inner choanoderm. Outer pinacoderm, it is made up of pinacocytes and porocytes. It is made of pinacocytes. Second one, porocytes. Pinacocytes are flat-shaped cells. Porocytes are tube-shaped cells. Next, inner choanoderm. It consists of choanocytes, which are also called as collar cells. which are also called as choanocytes. So, having these choanocytes is considered as unique characteristic feature in case of sponges or porifera. Choanocytes are unique characteristic feature in porifera. These choanocytes or collar cells have one flagellum and many microvilli. Now, the outer pinacoderm and inner choanoderm, these are having a middle substance called mesogia, gelatinous mesogia, which are having amoebocytes. We said that sponges are cellular level of organized organisms. So that these amoebocytes are of different types, that each and every cell is doing its own specific function. So between pinacoderm and choanoderm is present gelatinous mesogia with amoebocytes. These amoebocytes are of different types. You see a rough diagrammatic representation of sponges. Normally, sponges structure will be like this. These are labeled as ostia. Here will be innermost lining. We are telling this one as osculum. Inner one is made up of choanoderm or collar cells. In detailed structure of sponges. If we go for labeling, we can tell this is amoebocytes. These are choanocytes. This is mesoglia. These are spicules. And these are means the structures that supporting holding on to the substratum. These are basal tufts to hold substratum. So these are some of the important structures of the sponges, means the labelings of the sponges. So this empty space where these collar cells are releasing the flagellum is called spongiocy. Here the choanocytes constitutes the choanodere, and outer ostia it constitutes the pinacodere. Here gelatinous mesobilia, also called mesohype. Next, digestion in sponges is intracellular. You can also say sometimes digestion and excretion, respiration and excretion is by the process of diffusion. Respiration and excretion is by diffusion process. Next, pinacoderm or pinacocytes are flat-shaped cells or thin flat-shaped cells of epidermis. Next, choanocytes also means they also form into spermatocytes. Next, male and female gametes are produced in same organism, so sponges are also termed as hermaphrodites. Osculum is surrounded by myocytes. Next, coming to the structure, amoebocytes. Amoebocytes are of several types. First one, archaeocytes. They help in regeneration and nutrition. Next one, sclerocytes. They support in secretion of spicules. Next, spongiocytes. They secrete the spongin. Next, colencytes. Colencytes. They secrete collagen. Next is leukocytes, which support in means they try to move the organism from one place to another place, but as sponges are sedentary, that doesn't happen. Leukocytes, they are highly mobile. Next is phagocytes. They help in storing of food. Next one, trophocytes. They help in absorption of nutrition. Excretory material from sponges is ammonia. Reproduction takes place by two types, that is asexual and sexual type of reproduction. First one, Asexual type of reproduction is by fragmentation method. This fragmentation method or by formation of special cells called as structures called as gemmules or by special structures called gemmules. These gemmules have archaeocytes. Next, these gemmules are considered as endogenous buds also, are also formed as endogenous buds during sexual reproduction. Asexual reproduction. If you see the structure of a gemmule, how does it look like? So this will be the osculum or opening called as micropyle. So this is called micropyle. And here will be the spicules. Next, here will be the archaeocytes, group or mass of archaeocytes. These spicules are made of spongin fibers. Next, here I have written spicules in two different forms. So this is one single structure. These are called as monoaxon spicule. Such type of structures are called tetraxon spicules. This will be the outermost covering and this will be the innermost covering, which is holding on the archaeocytes. Archaeocytes, they form sperms and ova or male and female gametes. Sexual reproduction in case of sponges is internal and these are considered as hermaphrodite. Already we have seen that term. Sexual reproduction is internal. Next, sponges exhibit protogynous condition. It means that ova are matured earlier than the sperms. Next, fertilization is internal. Means the ova and or the male and female gametes between two sponges are exchanged so that there will be cross fertilization. Next, when archaeocytes forms sperms and ova, they are exchanged between two sponges. Fertilization is internal by water currents. We know that there is a water canal system. So forms zygote. After formation of zygote, in case of cycon and leucocylenia, there are larval forms. So cleavage happens from the zygote. In case of cycon and Leukocylenia, there are larval forms. Here, which is also called as scypha, the larva is amphiblastula larva. In case of Leukocylenia, there is parenchyma larva. Next. So this is about reproduction. Sponges based on spicules means what is the composition, chemical composition of the spicules or the skeletal system are of three different types. Sponges based on chemical composition of skeletal system or spicules are of three types. We can say three classes. One is calcarea, second one is hexactinellida, third one is desmospongia. So this is made up of calcareous spicules, and here it is made up of siliceous spicules, and here it is desmospongia are also made up of spongin fibers or siliceous spicules. But here it is 6 rows. Here it is 1 to 4 rows of siliceous spicules. These are few important points regarding the sponges.